we talk about biomolecules, we talk a lot about monomers and polymers. Monomers are the building blocks that are often repeated and can form a polymer. For example, with proteins, monomers can be amino acids. The polymer can be a polypeptide. But you know a question that used to keep me up at night? How do the monomers stick together? Like, how do amino acids join together to make a giant polypeptide? How do monosaccharides join together to form a polysaccharide? And then I learned about dehydration synthesis, also can be called dehydration reaction. You know, the word dehydration often makes me think of not having enough water, being dehydrated. Dehydration synthesis will involve losing water. And synthesis, if you remember, means to make something. So basically, you're making something and losing water to do it. Dehydration synthesis is a chemical reaction that, with some exceptions, joins many types of monomers together. Let's get into it. We'll start with monomers of proteins, amino acids. We know we usually draw amino acids like this, but let's get a little more fancy. Let's draw them showing the general elements in their structure. Remember we mentioned a popular mnemonic to remember major elements in biomolecules. Using that, you should expect elements like carbon, hydrogen, oxygen, and nitrogen to be found in proteins and in their amino acid monomers. And you'd be right. Here is one amino acid with its elements. On the left side, this is an amino group. Notice the nitrogen, the hydrogens. Here is a central carbon, also called an alpha carbon, and attached to this central carbon is a hydrogen and an R group, sometimes called a side chain. R groups can vary depending on the particular amino acid. There's quite a few amino acids to choose from after all. Now let's look at this on the right side of our illustration. It's a carboxyl group. It's got a carbon and oxygen double bonded here. That's a carbonyl, and it's got an OH. The OH is hydroxyl. Whole thing is a carboxyl. You know, just a side note, looking at this, the word amino acid might make more sense now because you see it's got an amino group and a carboxyl group. A carboxyl being acidic, so amino acid. Dehydration synthesis means that the hydroxyl, OH, of one of the amino acids on the carboxyl side and a hydrogen, H, on the other amino acid on its amino side combine. Water, H2O, is released, a covalent bond between the amino acids is formed, and in proteins, this is known as a peptide bond. Let's do another example. Carbohydrates now. We like to draw general monomers of carbohydrates called monosaccharides like this. But let's get more fancy here. Let's draw them showing the elements in their structure. If you remember the popular mnemonic for biomolecules, you would expect carbon, hydrogen, and oxygen in carbohydrates, and also monosaccharides, their monomers. And you'd be right. Here's the monosaccharide glucose. Beautiful ring structure. Here's another monosaccharide, fructose. Put them together and you get sucrose. This can form from a dehydration reaction. A hydroxyl OH from one of the monosaccharides, we'll show it coming from glucose, combines with hydrogen H from the other monosaccharide, we'll show it coming from fructose, and water is released. A covalent bond forms between these two monosaccharides. Specifically, the bond is known as a glycosidic linkage, or glycosidic bond. So both of these showed dehydration synthesis, joining these monomers together. But we feel there's a little bit of an elephant in the room, and that is, how did it happen? How did it start? Generally, this requires an investment of energy, and reactions are usually coupled with other cellular processes to make it happen. You can explore some details with that in our description. And to speed up the reaction, cells often have the help of enzymes or molecules that act like enzymes. Enzymes can also assist in making sure monomers are in position. One last thing. What you make, in this case, you can break. Sure, dehydration synthesis might join monomers to make polymers, but you can break them down again. The chemical reaction that breaks the bonds between monomers is called hydrolysis. Like its name would suggest, with hydro, it needs water. Take our example with the carbohydrate monomers that had been bonded together in dehydration synthesis. If a water molecule, H2O, is inserted in this bond that holds these monosaccharides, specifically in this glycosidic bond, you will notice that one monosaccharide gets a hydrogen and the other gets a hydroxyl. Enzymes often play a role in hydrolysis too. This particular example could involve the enzyme sucrase. So we went through dehydration synthesis and hydrolysis, and you might wonder, why does this matter? Well, biomolecules, like carbohydrates and proteins, perform essential functions for living organisms, and the different forms that many biomolecules take are critical for their function. Dehydration synthesis is involved for many, though not all, of these large polymer forms. And as for breaking them down, in order for organisms to survive, they are breaking down biomolecules. For example, take the human digestive system. Hydrolysis breaks down most biomolecules, breaking these down in digestion into smaller components that can be absorbed and used by the body. Explore all the diverse biomolecule functions in our biomolecules video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.